Hi friends, today I am going to discuss about the topic inhibitors of DNA replication, transcription and translation. First, let's look into central dogma. Central dogma shows us the flow of information from DNA through an RNA intermediate, finally forming proteins. We know that proteins are very important in all biological processes, and the central dogma is shown in three different steps, that is replication, transcription and translation. All these three processes should take place in a sequential manner such that the final end product will be the required protein, and if it is not taken place properly, the formation of protein will be seriously affected that can cause biological damages. DNA replication. DNA replication takes place during the S phase of cell cycle. DNA replication process is done by copying of a genomic DNA so that the progeny cells, that is the daughter cells, get equal amount of genetic information that was in the parental DNA. That is, parental DNA copies its genetic material so that the daughter DNA gets the same amount of genetic information after the cell cycle. Next one is the inhibitors of DNA replication. There are inhibitors for DNA replication, transcription and translation. First, let's look into the inhibitors of DNA replication. DNA replication process we have seen, and there are enzymes which take part in this DNA replication. They are known as DNA polymerases, and these inhibitors can either directly affect the DNA or can affect the functioning of the DNA polymerases. These inhibitors of DNA replication is divided into two categories. The first category includes the purines and pyrimidine nucleoside analogs. Analogs means these chemicals are structurally similar to purine and pyrimidine nucleosides, so that these analogs can replace normal purines and pyrimidines in the DNA, thereby affecting the DNA polymerase activity. The second category includes the DNA damaging agents. These can directly damage the DNA, which can affect the structure of the nucleic acid, thereby inhibiting DNA synthesis. The second category of DNA replication inhibitors are subdivided into four different types. First one is the alkylating anti-neoplastic agents. Second one is the DNA polymerase inhibitors. Third one, nitrogen mustards. And the final, topoisomerase inhibitors. Alkylating anti-neoplastic agents. These alkylating anti-neoplastic agents are used to inhibit DNA replication. That is, these chemical adds an alkyl group into the DNA, thereby affecting the normal DNA replication. Alkyl group can be either methyl group or ethyl group or like that any other alkyl groups. The common examples are normustine, mitomycin C, and alkyl sulfonate. All these act as alkylating anti-neoplastic agents, thereby adding of a alkyl group into the DNA. These mitomycin C and alkyl sulfonate can cause cross-linking in the DNA that affects the DNA replication. Next is the DNA polymerase inhibitors. We have seen that DNA polymerases are the enzymes which have a crucial role to play in DNA replication. So if these DNA polymerases are affected, then the normal DNA replication will be obviously affected. And these DNA polymerase inhibitors are commonly used in cancer treatment. We know that the cancer process or formation of the cancerous cells is the increased frequency of mitosis. So if these DNA polymerases are inhibited in such a cells, DNA replication will not take place, so that we can reduce the replication in the cancerous cells. Examples of DNA polymerase inhibitors are acetylcholine and cytosine arabinoxide. This acetylcholine act as an inhibitor of DNA polymerase alpha and delta in the eukaryotic cells. And this cytosine arabinoxide is commonly used in chemotherapy medication, which is used to treat myeloid leukemia. And next one is the nitrogen mustards. Previously, we have seen alkyl anti-neoplastic agent. These nitrogen mustards, some of the nitrogen mustards also act as alkyl anti-neoplastic agents. So these groups also add alkyl groups But these nitrogen mustards have a speciality, that is these react with the two bases on the opposite strands of the DNA. We know the DNA is having two strands. So these nitrogen mustards act in the basis of both these strands. They act in both these strands and it causes a formation of a covalent linkage. And these covalent linkage yield the formation of an adductor. This formation of adductor block the replication process. The common examples of nitrogen mustards are gulfosamide and the chlorambucil. This gulfosamide uh, is a compound that is of mustard agent with a potential alkylating activity. As previously said, these nitrogen mustards also have the alkylating activity. Next one is the chlorambucil. Chlorambucil has a is also bifunctional. That is, it also acts as a nitrogen mustard and also acts as an alkyl antineoplastic agent. Next one is the topoisomerase inhibitors. These topoisomerase inhibitors block the ligation step of the cell cycle. We know that topoisomerase are also important enzymes which act in DNA replication. So if these topoisomerase enzymes are inhibited, it will also affect the normal DNA replication. And these examples of topoisomerase inhibitors are anthracyclines and fluoroquinolines. These chemicals inhibit the DNA replication and also sometimes the transcription process. The cell cycle is blocked during S phase and the mitosis as a result of topoisomerase inhibition. If this topoisomerase activity is inhibited, then obviously the cell replication will also be inhibited. Next one is transcription. Transcription is the process of formation of RNA from DNA. The process of making RNA from the DNA is called as transcription. This takes place in three different steps, that is initiation, elongation, and termination. Transcription takes place with the help of an enzyme, RNA polymerases. So there are inhibitors of transcription. These are molecules which can inhibit either the role of DNA polymerases or it can act on the DNA molecules from which the RNAs are formed, so that the process of RNA formation will be inhibited. There are also two kinds of transcription inhibitors. Transcription inhibitors type 1, which interact with the DNA dependent RNA polymerases and inhibit the RNA synthesis, and the type 2 is which react or interact with the DNA directly, which inhibit the RNA synthesis. Type 1. These are inhibitors which interact with the DNA-dependent RNA polymerase enzymes and inhibit the RNA synthesis. Examples are streptoligidin, rifamycin, and alpha-amanitin. These rifamycin is a bacterial transcription inhibitor, so that we cannot use this rifamycin as an inhibitor for eukaryotes, such that it acts only on bacterial cells. Then alpha-amanitin is a molecule which affect the rate of RNA polymerase 2 activity. That is, it can reduce the activity of RNA polymerase 2, so that formation of RNA will be seriously affected, thereby inhibiting the transcription process. Next one is the inhibitors which interact with the DNA and inhibit the RNA synthesis. These chemicals do not act as an inhibitor for RNA polymerases, but instead they directly act on DNA, thereby inhibiting the process of transcription. And these type 2 types of inhibitors are actually antibiotics. That is actinomycin D, chromomycin A3, and mithramycin A. These three chemicals directly interact with the DNA, thereby preventing the transcription process. These actinomycin D has the ability to inhibit the growth of rapidly dividing cells, so that it acts as an effective agent in the cancer treatment. Chromomycin A3 is also an important chemical, that is, it binds to the DNA at the minor groove and inhibit DNA transcription. These chromomycin A3 can also act with divalent metal ions and play a role in inhibition. So the activity of chromomycin A3 is related to the presence of the divalent metal ions. Then mithramycin A. These are DNA binding transcription inhibitor, and these mithramycin has a property to bind with the GC rich sequences, that is guanine and cytosine rich sequences, which are located in the minor groove of the DNA. These GC-rich sequences are usually located in the minor groups of the DNA. So methanogen C binds with these regions, thereby inhibiting transcription. Next is the translation. The process by which a cell makes protein is known as translation. mRNA is made by copying the DNA and the information it carries the cell, it tells the cell how to link amino acid together to form protein. Inhibitors of translation. The protein synthesis inhibitors are the compounds that stops or slows down the formation of proteins. We know that the formation of protein takes place with the help of ribosomes. The ribosomes are important cell organelles for the synthesis of protein. There are two subunits for the ribosome, that is 30S subunit and the 50S larger subunit. The 30S subunit is a smaller subunit. So the inhibitors of translation can either bind with the 30S subunit or with the 50S subunit. The examples of antibiotics which bind to the 30S subunit are aminoglycosides and tetracycline. Aminoglycosides are usually used as an antibiotic, which act as an which is an amino modified glycoside, so that it affects the normal translation process. These tetracycline are commonly antibiotics which also act as an inhibitor of protein synthesis by binding to the 30S subunit. We know the 30S subunit has a major role to play in protein synthesis than the 50S subunit of the mRNA molecule. mRNA strand is coming and attaching to the smaller subunit, that is the 30S subunit. And these tetracycline also inhibit the binding of amino acid tRNA to mRNA translation complex. Next one is the antibiotic which bind to the 50S ribosomal subunit, that is chloramphenicol, clindamycin, and linezolid. This chloramphenicol is an antibiotic which blocks the protein synthesis, but it will not affect the process of transcription or replication. And this chloramphenicol works in a mechanism such that it prevents the binding of the amino acid tRNA to the peptidyl transferase active site. We know that there are three important active sites, that is exit site, peptidyl site, and one A site is also present, that amino acid site. So this chloramphenicol prevents the binding of amino acid tRNA to the peptidyl transferase active site. This clindamycin inhibit the early chain elongation, thereby proteins are not formed of required length. Then next one is the linezolid. These linezolid also inhibits the translation process, and it is increasingly used in treatment process such as for gram-positive pathogens. These have an action of inhibition of bacterial protein synthesis. So usually used for prokaryotes and not for eukaryotes. As a conclusion, we have seen that these inhibitors have a major role to play in inhibiting the process of mitosis. These mitosis can be seriously have a major role in cancerous cells. That is, the cancerous cells are the multiplication or the rapid multiplication of normal cells. So the rate of mitosis in these tumorous cells are more than the normal cells. So if these mitosis in the tumorous cells are reduced, then these can act as a treatment of cancerous cells. So these inhibitors can be used for the treatment of cancerous cells, so that these can maintain the enhanced metabolism and proliferative cells of the proliferative state of the normal cells. These cancerous cells can be susceptible to transcription and translation inhibitors, so that it can be widely used for the treatment of a serious disease known as cancer. And these inhibitors are useful in such a way that can be used as a treatment process in the present scenario as the cancer is increasing. The number of people who are getting cancer is increasing day by day. So these inhibitors can be used for the treatment of cancer. These are my references. Thank you.